that is very near and dear to all of us here on this presentation platform. I'm going to give just a few very general remarks to get us started and oriented as to why an evolutionary lens is really important. And then I'm going to turn it over to Antonis and Tony to introduce their teams and to get the real meat of the presentation going. So as Charlie mentioned, I am a pediatrician by background. And the area of investigation that has really captured my attention is this one. It's understanding why babies are born prematurely. Preterm birth, which is defined in people as birth at less than 37 weeks of completed gestation, accounts for 10% of births around the world each year. 4 million deaths occur in the first month of life worldwide, and preterm birth is the single largest cause, whether you're in the United States, other developed countries, or in the developing world. And there has been little change in that number for the last 50 years, and it's particularly due to the fact that we don't have any generally effective interventions. One metric I like to use in terms of describing the impact of preterm birth compared to adult disease as we think about is this metric called the worldwide disability adjusted life year. So this is years of lost life or impaired life due to different disorders. And as you can see, perinatal disorders, the bottom bar, far exceeds other diseases that we think are important in the adult world, like cerebrovascular disease, heart disease, depression. And I think if we could make a little impact on these perinatal disorders, we will be doing mankind a huge service. So for us, the big question is, what in general determines the timing for birth? We know that every organism is born, emerges from the intrauterine environment at a very characteristic time. In humans, on average, this is 266 days from conception. If you're a mouse, it's 19 days. If you're a killer whale, it's a couple years. But how we know we're at that point is a mystery. Is there a clock that starts at the beginning of the gestation and then ticks off? Or are there signals that go back and forth between the mother and the baby? And then when you think about preterm birth, the situation gets even more complicated because there's all of the usual physiologic factors, but then there's all the environmental factors that overlay on that, like infections, stress, socioeconomic influences, et cetera. So disentangling this really becomes a big problem. And then in terms of thinking about complex common disease, it gets even more challenged by thinking about the genetics of pregnancy. It involves 2 genomes, the mother's genome, and 3 haplotypes to confer this shared multi-genome of pregnancy, this metagenome of pregnancy between the mother and the infant. So disentangling what is due to fetal genotype and phenotype and what is due to maternal genotype can be a challenge, and especially since they share genetic information. So one typical way in the past that people have made progress in understanding complex biomedical diseases is to use animal models. And unfortunately, for human pregnancy, there really are no good, easily manipulatable animal models. The mouse, an organism I've used a lot to study pregnancy, by even the grossest looks, is very different from human pregnancy. The shape of the uterus is different. It's bicornuate as opposed to piriform. All mouse gestations are multiple. They won't maintain a singleton gestation pregnancy from conception. And then progesterone, the hormone that maintains the uterus in a quiet state, comes from the mother system, the ovary, throughout gestation in the mouse. Whereas in human pregnancy, 97% are singleton, and the progesterone shifts from the ovary in the mom at 10 weeks gestation to the placenta. So the hormone that controls gestation is actually coming from a different individual than it is in most other animal model systems. So that has made the situation challenging, at least, and very difficult to unravel with animal systems. So about 15 years ago, we wanted to test this hypothesis, that genes involved in preterm birth are rapidly changing in humans. And we postulated that because human pregnancy has to accommodate species-specific adaptations, most notably a large head and a relatively small birth canal size that is necessary to facilitate bipedalism. So our prediction is that delivering the human fetus at a relatively earlier gestational age, still mature enough for survival, will be beneficial. And a corollary of that would be that you would predict humans are relatively immature at birth compared to other primates. And if you compare us to our nearest primate relatives like chimpanzees, chimpanzees are born with much more developed dentition, much greater brain development, including developed vision, suggesting that this is indeed the case. So we know now, in looking at the human and chimp genomes, that we only differ in terms of nucleic acid sequence in about 1.5%. But we know that 1.5% is extraordinarily important because it's led to this massive increase in human cranial capacity and brain size, shown here, increasing about threefold, which is great because now we can come on Zoom and talk about human evolution and birth timing, but it actually made some challenges for birth. So here's a picture of the obstetric canal. This panel shows the baby's head emerging cranium down outside the birth canal. And you can see in this lower panel that there's not a lot of extra room of the baby's head at birth compared to when it's delivered at term. If you look at Neanderthals, different in evolution from us about 120,000 years ago, the head was the same size, but the birth canal shape was different. It was much more open and didn't have the same kind of constraint. And then if we look back through evolution, chimpanzee 5 million years ago, Australopithecus about 2.5 million years ago, and humans, we see this progressive shrinking of the obstetric capacity, the size between the brain and the birth canal. So that could be one driver of birth timing. The other alternative hypothesis was published by Holly Dunsworth and her colleagues in PNAS a few years ago now, called the metabolic crossover hypothesis, where the baby's energy demands exceeds what the mother can supply, and that sends essentially a starvation signal to the mom to deliver the baby. And it's probably all driven by the brain is kind of the bottom line, but we really don't know how it does that. So several years ago now, with Justin Fay, a colleague who is at Washington University, we did a comparative genomic screen looking for genes that were rapidly evolving along the human genome as potential candidates as one strategy. But I think what you're going to hear about today is a much more sophisticated look at how evolution has influenced birth timing and what the constraints on evolutionary models are. So with that preamble, I am going to turn it over to Antonis to introduce, and Tony to introduce their team who led the efforts around the paper that Charlie described to you.
Hi, everyone, and thank you, Lou, for the wonderful introduction. Yeah, this is work that was led by by people in the labs of Tony Capra, who was as recently as a couple months ago at Vanderbilt University, but very recently moved to the University of California in San Francisco. And also Patrick Abbott, a colleague at Vanderbilt, was also instrumental in developing some of the ideas. But the real stars of the show are two people in Tony's group and one in my group. And these are Abbe LaBella. She did her PhD at Duke University. So some of you may be already familiar with her skills and talents. And she works in my lab. And then Abin Abraham, who is an MD PhD student in Tony Capra's lab. And the two of them hit it off. And anything that's wonderful about this work is really thanks to their contribution. So I believe Abin is going to go first, and then Abby, and then we'll have questions. Thank you. Abin, take it away.
Awesome. I'm going to share my screen here. Can everyone see my slides here? Awesome. Thank you for introducing us, Antonis. I'm going to get started talking about our analysis on genomic regions associated with spontaneous preterm birth. So a lot of our work derives from this genome-wide association study that was published a few years ago, thanks to the leadership of Ge and Liu. And this GWAS is the largest GWAS we have looking at spontaneous preterm birth. So many of you are probably familiar with this Manhattan plot, but each point here represents a variant in the human genome that was tested for an association with spontaneous preterm birth. On the y-axis, you can see the log 10 p-value. And so points that are higher on this figure show a higher strength of association with spontaneous preterm birth. We often use this genome-wide p-value threshold to correct for multiple testing. And when we apply that, there are a handful of regions that look like potential candidate regions associated with preterm birth. So when we start with this Manhattan plot, and we ask this question, how do we detect selection in the human genome? And very broadly, we rely on patterns that are left behind by actions of evolutionary forces. So for example, here you can think about each horizontal line representing one genome or one person, and these black dots represent different variants in the genome. So collectively, this represents our population. And when we have these 3 red variants that have some beneficial effect on fitness, they're going to be positively selected. So over time, what will happen is more people in this population will start to have this particular allele at this variant. And as even more time goes on, it will reach fixation, or everyone in the population will now have this allele. So this pattern that you see here is what we sort of expect from positive selection. And this is just one example, but the point I want to emphasize is a lot of the way we try to detect selection depends on the signature or the patterns left behind by these evolutionary forces, which you can see in this panel is the variants are far more structured than what we originally started with. So that was an example of positive selection, but certainly we can describe selection in terms of negative, balancing, or local adaptation. And thanks to work from various labs, we have measures that can look at those signatures left behind these selective forces and have a way to quantify them. So here we're going to look at PhyloP, FAST-ON, SCRP, and Linsight, and these give us an idea about positive and negative selection. Likewise, we can also infer the age of the allele using coalescent models and various other techniques. We also have these integrated haplotype scores that give us an idea about positive and more recent selection, as well as beta score, which was more recently developed looking at balancing selection, and finally FST that gives us an idea of local adaptation that occurred in the human population. So our goal was to start with the genome-wide association study, identify independent regions, and then try to detect different evolutionary forces that have acted on these regions. So I'm going to briefly go over the details of how we do this, but please ask any questions if you have them. From that Manhattan plot I showed you, what we do is identify independent regions. So here is a schematic of a region from that Manhattan plot. These circles and diamond represent variants in that plot. And you can see we're going to call the variant with the lowest p-value the lead SNP. And once we have our region, what we do is match that region based on minor allele frequency and the linkage disequilibrium structure to 5,000 different sets. And then we want to measure selection. So we take one of those evolutionary measures I mentioned earlier, and we see what are the values in the region, and also what are the values in our match set. And to summarize that, we're going to take the median across our region of interest and all of our match regions. And finally, we can ask, how does the region that is associated with our trait compare to this matched background? So by looking at this empirical distribution, we can derive a p-value and a z-score. So in our analyses, one of the things we found out was that the choice of the background distribution matters. Often in many genome-wide selection scans or other previous approaches, people often look at the genome-wide distribution. However, we know that minor allele frequency and linkage disequilibrium are important factors that affect those signatures that we see. So here we're going to show 4 representative representative regions to illustrate why the choice of the background distribution matters. Here we're looking at this region where the black distribution represents a genome-wide distribution of this evolutionary measure of FST. The orange distribution represents our matched set of regions on minor allele frequency and linkage disequilibrium. And here I am demarcating the 95th percentile. So anything past these guides essentially represent a p-value less than 0.05. You can see in this example, this particular region, it would be called significant if we use a genome-wide distribution, but it would not be significant when you look at the match distribution. And that's what's being summarized on the table on the right. So here are some other representative examples. And in these three examples, you can see the black line is the exact same throughout all four, representing the genome-wide background, but the match distribution has very different shapes. And as a result, what we call as significant varies. So going back to our selection pipeline, what we do is start not just look at those four representative variants, but we identify all the regions associated with spontaneous preterm birth. And just as a note, we reduce this genome-wide threshold because it's very likely that many of these regions just did not reach statistical significance because of power. So for example, if you increase the sample size, you're way more likely to find more regions. And I'm going to orient you to the results of this selection pipeline. Here is a heat map where each column represents a different genomic region, and each of the rows represent the evolutionary metrics that we talked about earlier. They're categorized by the type of selection that they describe. And the color and the size of the squares represent the z-score derived from that empirical distribution I mentioned earlier. We've also annotated in a star those regions with a p-value less than 0.05. So when you look at all the regions we were able to identify from that GWAS associated with preterm birth, what emerges is this particular heat map. We have clustered the genomic regions based on their patterns of evolutionary measures that we looked at. And then we're able to infer the types of evolutionary signatures at each of these locations. And what we noticed was it's not that there's one dominant mode of selection that is acting on the genomic regions associated with preterm birth. And with that, I'm going to hand it off to Abby, who will describe a little bit more details about what we learned from this analysis.
All right, thank you so much, Abin. I'm going to take over discussing some of the things we found when we really delve into the particular variants that were under investigation. And so these are the ones that we covered within our paper. You can see that we picked a representative area for each evolutionary process and then looked at what the gene and the variant are that were associated with this GWAS. And so I'm going to go over some of the more interesting results. So I'm going to cover two. The first is the GC gene, and the second is OPRL1. This OPRL1 fell within this group that we called diverse just because there was no single dominant signal that we could see, but I'll discuss what we detected when we get there. So first, I'm going to talk about a variant that we found within the intron of the GC vitamin D binding protein. Here in red, you can see the lead variant that we saw with the lowest p-value in the GWAS. And then here you can see the other linked variants in orange that were also intronic that were linked to this lead variant. I'm going to talk specifically about the lead variant in this analysis so that we can learn a little bit more about what's happening. So this lead variant has two alleles, a protective and a risk allele, and it appears that the risk allele has emerged in the human population and is not found deeper within the great apes. And we actually do have some population level data from the great apes, and it was not variable at this position either. We can also look at the haplotype for this region. And so what we see here is that the protective haplotype is most common in the East Asians and European population. But when we look at the risk haplotype, we can see that it's dominant in the African population. And so even though this GWAS was done in Europeans, when we look at the evolutionary history of this variant, we can see that the risk allele is more prominent within the African population. We also look to see what other data was available in GWAS for this variant, and we see that the risk allele is also associated with another disorder where we see an increased risk of arthritis in a spine condition. So this is to say that we do see some evidence of local adaptation within this region that that was detected in the SPTV GWAS. Many of you are probably familiar with vitamin D, but when we look within the literature, we see that vitamin D levels prior to delivery are associated with spontaneous preterm birth. We can see that levels of this actual protein within the cervical vaginal fluid may actually help us predict those that are at risk for spontaneous preterm birth. And this differential population structure within this region is really associated with the idea of differential evolution of the vitamin D system within populations. So our evolutionary analysis is really in line with the thinking about the vitamin D binding protein's role within gestational duration. So now I'm going to move to the second region that I wanted to talk about and share. This is a variant located within the 5' UTR of the OPRL1. And this variant was flagged because it had a very strange haplotype structure detected by the IHS within some populations. And what we can see is the opposite when we do a comparative genomics analysis, in that this protective allele appears to have arisen within the human population, but the risk allele, obviously risk in humans, is not found within the rest of the great apes. And as you can see from the haplotype network for this lead variant here, there was only one variant in this region. You can see this sort of crazy risk haplotype structure. It's very clear that this protective allele likely arose recently, has a very solid haplotype structure, not very complicated. There is, a weird thing where we see the emergence of the risk or the protective allele coming in a European population from a separate haplotype. And what you can also see here is that a lot of this structure is driven by haplotypes, a complicated haplotype structure within the African population, all of these down here are orange. And when we look at other information, we see that this variant is EQTL for the gene OPRL1 and another gene here within multiple tissues in the human body. So it really looks like we are getting to something that has to do with expression. We can see that there's changes in whole blood, brain, aorta, heart, and esophagus. So this is the data that we see that's really consistent with the evolutionary signal that we see for a complicated haplotype structure within the African population, and a more simple one within the European and East Asian. And what is OPRL1? What does it do? So in this data, when we looked within the literature, OPRL1 encodes this nociceptin opioid peptide receptor. It's a G-coupled protein that binds to nociceptin. And essentially, nociceptin and pre-nociceptin are associated with contractions and relaxation within the uterus and can be associated with preterm birth. So what we're seeing here is likely a region that's associated with the onset of contractions, so the onset of delivery, and may have a differential population structure within the different human populations. So this is the type of analysis that we did, looking into what are the function of these regions. Does our evolutionary analysis make sense with what is known? And how can we add to the thinking about how these regions contribute to spontaneous preterm birth? So I covered just two of these little vignette stories, but if you'd like to see the complete story, the paper was sent around, but here's another link for that information as well. So what you might be thinking, and what many of the reviewers of the paper were thinking, was, well, why did you just focus on spontaneous preterm birth? How can we apply this to other disorders? And so I'm going to go into a little bit of the ongoing work that we're doing now This is all preliminary, coming hot off the presses. And this was motivated by what are other traits mosaic in their selection? And so now I'm going to shift focus from a analysis on individual genomic regions to analysis on the trait as a whole. And one of the questions that we can ask is, how does spontaneous preterm birth with its multiple genomes in 2 tissues compare to traits such as HDL levels or other disorders and traits within the human population? And we're really looking at what is the selective pressures or landscapes that are associated with these traits. And one of the reasons that we can do this is that there are hundreds of GWAS available that currently have the appropriate data that we can deploy to pipeline. And so what I'm going to do is show you what this trait level analysis looks like. We call this the selective architecture, and these are the same metrics along the outside of this radial plot. We can see those associated with conservation, those associated with population differentiation, haplotype structure, allele. Is there a question? Okay. And so what we see here is that this is sort of no enrichment within these metrics. Anywhere where you see a dot above this central line means that we saw an enrichment in this metric on the trait as a whole when we looked at all the variants together. Anywhere below this line, we see a depletion. So the value was lower than what we would have expected when we looked at a matched control set. And anything in red is a p-value less than 0.05. So what we're seeing here is the selective landscape associated with the variants for the trait gestational age. So this was from the same GWAS paper as the SPTV GWAS, but this is just a little bit more interesting of a result to share. So this is what we're calling, again, the selective architecture of this trait, and we can use this to compare between traits. And so here we're looking at the radial plots for gestational age on the left and HDL levels on the right. And what you can see is that there are some different patterns within this. And one of the goals of our ongoing research is to ask questions about what are the selective architectures between different types of traits. And so I'm just going to narrow in on one region within here, and you can see that there are different patterns of population differentiation acting on the variants associated with these 2 traits. And so we're working on expanding this analysis to lots of different traits. Here we have educational attainment, HDL, LDL, BMI, weight to hip ratio adjusted BMI. And so we're really excited that we're going to be able to deploy this pipeline on tens to hundreds of GWAS to really start asking questions about how the selective architecture of these traits varies with things like heritability or penetration, to see what that looks like. And if you're interested in deploying this pipeline, all you need is a GWAS. You can go to Abin's GitHub, and you can see the link here, and you can download and deploy this pipeline. I've been running it day after day. It's very easy to use. So we hope that you will take a look at this pipeline, and you can even add in your own evolutionary metrics. It just has to be in a bed file. And with that, I want to thank everybody who is involved in this project. Obviously, the project leaders that you've talked to here and all of the funding sources. I can't wait to get a new picture of the Capra lab and the red haziness out in San Francisco. And with that, we're super happy to take questions or comments. You can always reach out to Abin and I by email, me on Twitter. We'd be happy to answer any of your questions about this work, the paper, the pipeline, and we have plenty of time for discussion. So we are excited to hear from you.
Excellent, that was great. Fantastic, thank you for that great presentation. How should we handle things from here? Did anyone else on the team have anything they wanted to say? Antonis or Tony or Lou? Should we just
I've just been following the chat discussion, which has been great. And a lot has come up around the obstetric dilemma hypothesis and sort of concerns around it. And I fully appreciate those. Didn't have a lot of time to go into nuance and discussion points there. But, you know, there's not great evidence that, that the pelvis couldn't evolve to accommodate a bigger head size and still be perfectly capable of very effective bipedalism. And the metabolic crossover hypothesis, I think, is very attractive. The thing we do know is It's probably about the brain. Humans are delivered when their brain is about 25% of final size. Chimpanzees are delivered when their brains are about 40 to 45% of final size. So, I mean, the human brain is just not as mature. So there is this sort of sense that things are pushed a little bit earlier, either to accommodate metabolism, size, or other parameters that we don't completely understand.
That's great. Would anyone like to raise any points about that while we have that topic on the floor? Are these hypotheses mutually exclusive, for one?
Hi, my name is Saran Weir, and I'm actually an obstetrician. I have a quick question. Do we know in other primate species, like in chimpanzees where that's been brought up, do the chimpanzee mothers experience the same obstetrical complications that human mothers experience? So a lot of the problems as women are pregnant longer are things like preeclampsia and other maternal complications. So I'm just wondering if there are corollaries in the chimpanzee population, and is this maybe driven by the maternal space as opposed to the fetal?
Yeah, well, in all of the studies that have been done so far, the vast majority of genetic determination of birth timing is in the mother. Fetal size is mostly programmed by the fetus. And then for preeclampsia, the existing GWAS hit is in the fetal genome in SFLT1. So in terms of other primate models for preeclampsia, there really aren't any naturally occurring ones. Other species don't have the same constraints. But chimpanzees do have preterm birth, as I think, or other primates do have preterm birth. Antonis, I don't know if you want to comment on the data you reported a couple of years ago.
Yeah, a few years ago, we wrote a paper in EMPH, of all places, about whether preterm birth is unique to humans, and it's clear that it's a quantitative trait, and and in humans it's defined as 37 weeks of completed gestation, or 92.5% of completed gestation. And so if you apply that rule, then you find that virtually any outbred organism has preterm birth. What I think differs is the phenotypic consequences. So because of the immaturity of the brain that Lou is talking about, I think what differs between, so in other primates, if they give birth prematurely, if it's very early prematurely, then there are issues with the baby. But if it's just a few weeks before, then the babies seem to be doing fine. They don't seem to have any overt phenotypes. But I should add the caution that we desperately need studies like that. If you have access to zoos, or if you have access to populations like those, and you could characterize that, you should go and do it. You would have a reviewer who would be very favorite in me.
Is the paper you're referring to in EMPH, that was the one on dogs?
No, there was an early one, 2015. I'll just post it as a link.
I'll paste it in. I think I know which one.
It looks like we have a couple hands up. Dr. Graves?
Yeah, guys, if you were looking in the chat, then you saw that I made a bunch of comments. I'm long been known as extremely critical of these kinds of research approaches. When I give my EvMed talk in a couple of weeks, I'll have an opportunity to explain in greater detail why I think these kinds of research approaches don't work. But there were some questions that I had right off the bat that the presentation didn't seem to answer, and the one that really needed to be heard, which is number one, the GWAS studies that was reported on preterm births in Nordic populations, what percentage of the phenotype do they explain?
If you do GPTA analysis across all of the SNPs in the study, it explains about 25% of the risk for preterm birth. If you look at the ones that achieve genome-wide significance, it's only about 1%.
And by the way, that's typical, because when we talk about exactly.
I would argue they are pointing to pathways right now that we never envisioned before as being even remotely involved. I mean, the Wnt pathway makes perfect sense, but we never thought about it. So to me, it's a way of getting your foot in the door in an unbiased fashion into GWAS studies that you then begin to take apart in terms of epigenetic modifiers, in terms of how environment modifies those signal pathways. But we don't have a way to really study human pregnancy in a genome-wide, unbiased fashion without doing similar things.
Well, Lou, I just want you to know that I publish these GWAS studies myself. I do it on lifespan in fruit flies.
Mm-hmm.
So I'm very familiar with the methodologies that are used, and that's why I make this critique of exactly, what level of significance we're talking about and how much does it actually explain the phenotype in question, which leaves huge questions in terms of the overall experimental design. And one of the fallacies that, again, I've continually pointed to in these kinds of studies is the fact before you can assign any genetic causality to any variant, the populations that are being studied, your cases and control, have to be existing in a shared environment. And usually that shared environment should exist for at least 2 generations before you do the study. And that never happens in these human GWAS studies at all. And so for me, much of what we get out of them, could be true, but it really sort of fails to rise to the level of strong inference that we use, at least in my field of genomics.
Well, just to counter that, we did our studies in Finland just for that reason, because it's a genetic isolate. It's been a stable population for many years, and that was a motivation for doing that. But we've repeated this analysis now on populations from Africa and from South Asia and from China, and the same hits come up in all of those as well. We've expanded the data quite a lot, and so far, it's very consistent. Does that mean it's proof positive, or that it has helped us understand mechanism yet? No, we need to go into now. To me, the limitation with GWAS is people stop at GWAS, right? They say, eureka, discovery, we've hit the jackpot, but then they don't figure out what mechanism is. Most of the time, you don't know what the gene is, you don't know how it's acting. And to me, that's where you really have to do a deep dive, if you believe the hit, which we do.
Okay, I want to call on Herman Poncer. If you're still here, Herman, and have a question, I see your hand is up.
Yeah, thanks. Great talk. I don't do GWAS work, so I don't have anything to say about that. But one thing that Lewis mentioned as he was introducing it, and I think is something kind of central to this, is the idea that the human brain size being so big might be a big factor in driving gestation lengths. And I totally agree. I'm on that Dunsworth paper looking at metabolism, so I'm a biased commenter here. And I'd like to see more work done to try to disentangle obstetric dilemma arguments and hypotheses versus metabolic. One thing I just wanted to add and say about the brain piece is that actually brains, human neonates have brains that are bigger in terms of absolute size and bigger in terms of moms as a proportion of mom's body size than chimps when they're born. So somehow mom's getting all that extra resource there to grow a bigger brain. Now it's a smaller as a percentage of the adult size because our brains get so huge, so the denominator there ends up being really big.
Exactly.
But as we search for these mechanisms, and that's my comment. My question would be something that I think Charlie put in the chat. Are any of these either GWAS inferences or other lines of evidence, if we don't like GWAS, pointing towards perhaps something to do with skeletal growth, which might point to obstetrical dilemma issues versus, glucose metabolism or fat metabolism or something like in those pathways? Are we able to maybe sort out some of those to begin to get some traction on this debate?
Well, maybe I can just start. The problem is most of the really high level hits that we've gotten are involved in so many different pathways that it's hard to know. So one of the hits we got was in Wnt4. Wnt4 has already been demonstrated to be a GWAS hit for bone density and bone growth. So and it's also involved in uterus development. So which of the two pathways is it? EBF1, our top hit, is involved in immune system regulation, but it's also highly involved in a bunch of cardiovascular disorders. It's been associated. So the pleiotropy is just really daunting as well.
Yeah, this is Go from Cincinnati. I work together with Lou in conducting that GWAS study. To answer your question, one of our top hit is ADCY5. That's clearly blood glucose loci.
Great, thank you.
That's helpful. That's cool to see.
All right, so I believe next we had a question or a perspective from David Haig. David, do you want to go ahead with your comment?
Okay, so it's the conversation has passed on a long time since I was wanting to put a comment in, but I think on talking about human birth as occurring earlier relative to brain size, I think it's more convenient to think of it as that human infants are delaying the maturation of brain size. The human pregnancy is longer than the chimp pregnancy. The really distinctive thing about human babies at birth is just how incredibly fat and how much white adipose tissue they deposit. That's more anomalous actually than brain size at birth. I think one thing that's happening in humans, and it comes under the general label of secondary autritionality, that human infants have evolved to be more so-called helpless and to delay a lot of development. Curiously, autritionality is also associated in nakedness and in mammals that produce litters. And there's a certain sense in which humans are litter-producing animals, in that, unlike a chimpanzee, which raises an infant to independence before the mother has the next child, we have overlapping care. And so I think the evolution of autritionality, to understand it, and with that is the delay in brain development, has to be put in the context of inter-sibling interactions and their interactions with the mother.
Very interesting. Well, the notion of, the adiposity that human infants have at birth is just it's different very strongly than any other even ape. And what are your thoughts about that? I mean, what is there a link to the brain?
The best thought I've had on that particular one is that a pound of fat in the baby is better than a pound of fat in the breast, other things being equal. And I think, so for a chimpanzee infant, when it's born, if its mother is not able to get up immediately and care for herself and care for the infant's in trouble. In humans, we've got social care at birth. People are caring for the mother and for the infant. And so that relaxes the demand that infants are making on the mother. And so it's switched the balance to them, so mothers can have a Wochenbett, a week in bed when they're not out and feeding, and the infants still survive. But that's just a theory.
Can I just jump in for a second? Human adults are also much fatter than any ape adults. So chimpanzees, even in a zoo, carry about 10% or less body fat. And that would be, Olympic level athlete body fats in humans. And so even in hunter-gatherer groups and other subsistence level economies where people don't have access to supermarkets, women carry, in the 20% of 20 is in terms of body fat percentage or more. Men are higher too. So it's interesting because it's not just that the babies are born with all this fat resource. The women, their moms are also, by being humans, are also going to have a lot more fat. So both of those things have happened. We don't know the timing of them, but right now they both co-occur.
It's interesting too that EBF1 is the tissue it's most highly expressed in is the adipocyte. And it's in our genome LD block is an EQTL for EBF1 in the adipocyte.
Okay, I want to call on Karen Rosenberg for the next question. Karen, are you still there? And would you like to ask a question?
Yeah, thank you so much. I had a couple things I wanted to say. First, I totally agree with Charlie about the idea that these alternative hypotheses are not mutually exclusive. So this, the metabolic hypotheses that Herman and Holly proposed are certainly not contradicting any ideas about obstetric constraints. The second thing is that we don't know so much about birth in non-human primates, especially in the field, because non-human primates usually give birth on their own. They try to get away from potential predators. They try to get away from conspecifics and from primatologists. And so it's hard to observe those. But we do know about birth is that chimpanzees are probably not a very good model for humans. The constraints on obstetrics in monkeys are much more similar in terms of the space that's available as the head passes through. And although we don't know anything about, obviously, things like pain during labor in non-human primates, because we can't ask them about it, it seems like monkeys have a longer, prolonged labor, more like humans, and chimpanzees seems to go fairly quickly. But there have been a few studies recently in labs observing non-human primates, and I think those have really complicated the issue in really interesting ways. Just two more things. The other thing I wanted to say is that there might be other sources of selection besides energetic efficiency on the pelvis that might be balancing selection from obstetrics. Some people have proposed that injuries, likelihood of injuries might be important. Women have a lot more knee injuries than men, and that might be the result of the geometry of the pelvis. I think it's interesting. I've just sort of started thinking about pelvic floor musculature and the morphology of the bony pelvis. So I think there are other possible things besides just energetic efficiency. And then the last thing I wanted to say was about the term altriciality. So humans are not really altricial, right? So when Portman said that, I mean, obviously primates are precocial, small litters, long gestations, blah, blah. Portman proposed that humans were secondarily altricial because we were more helpless at birth than other primates, but we're not like mice, right? So in a way, I think that it's kind of a little bit misleading. So that's what I want to say.
Okay, thank you, Karen, for sharing that perspective. We're almost at the end of the hour, and so before we stop, I just wanted to circle back to Abbe and Abin and just see if you had anything else you wanted to add in relation to the conversation, or if you've seen anything in the chat box that you think you'd like to respond to or highlight. Abbe, do you want to start?
Sure. I think sort of along the conversation of how much can we explain with GWAS? What are we doing with all of these GWAS? There's GWAS on clapping beat. We are really sort of trying to detangle like what we know from what information is out there. And we're very cautious within all of our manuscripts and hopefully in the way we speak that we aren't trying to say, this is the be all end all of how HDL evolved in humans, but we are trying to use the abundance of data out there thoughtfully and cautiously to start to disentangle some of these questions. We didn't find a GWAS variant that was under, human specific evolution that was also strongly associated with waist to hip ratio. So we really think that we are chipping away at creating hypotheses that we can test and look at other organisms by trying to take the GWAS data and really apply an evolutionary lens to it. And so I think that that's just what we're trying to maybe provide a little bit of evidence for some of the ways in which the evolution has shaped the variants that we know about. So I just hope that, we're not trying to say this is the evolutionary history of this trait, but there is a lot of data out there, and for the most part, it hasn't been looked at through this evolutionary lens. So with that, if Abin has anything else to add.
That was great. Abin?
Yeah, I'll definitely echo what Abby said, and I really appreciate all the evolutionary perspectives that we discussed, and also in the chat. I think it helps us sort of focus our hypotheses, and all of this is very fascinating. But thank you for having us today.
Absolutely. Thanks for sharing, and I'll now do the same with Tony and Antonis. If you guys have anything you'd like to say as we wrap up.
Yeah, I think I'll echo all the thanks, especially. This is an amazing group, and I've really enjoyed getting to hear everyone's perspectives. And I think it's important to highlight that while these GWASs, for preterm birth and for most other complex traits, are not providing us with the one gene for that trait that we're going to go in and edit the genome and change and solve the problem. As Lou's work has been showing, they are providing insights into what are the pathways, what are the relevant systems that are involved in those traits. And now we're trying to layer on top of that the evolutionary histories that have shaped those pathways. And I think the biggest weakness here is there's still a lot of the genetic influence on these traits that we do not see. And as we understand more and more, I think that that will also allow us to sort of more confidently test some of the hypotheses like those have been raised here today. So it's great for you to share those with us. And again, just big thanks to all of you. And also want to mention that Abby is on the faculty market right now. So if anyone knows of a job out there and would be interested in hearing more about what she wants to be up to in the future, please reach out to her.
Thank you, Tony.
Great. And Antonis, did you want to say anything? Respond to any comments in the chat box or provide any last perspective?
Yeah, so I agree with everything that has been said, and I see your criticisms, and I definitely do hear them. In my mind, what we're trying to do is, right, a lot of the work on pregnancy and medicine in general has been phenotype driven, right? And there are a lot of hypotheses. And so in the last 20 or so years with the revolution in genomics, we have sort of going in the opposite direction, right? From genomics to phenotype. And I see these approaches as complementary. And, in many ways, and I think we've talked a lot about this with Abby and Abin and Tony and Patrick, it's more important to ask the questions and to sort of probe and, chip away at the, what the results may be, as opposed to sort of what the result is that we're getting. And so we're definitely into the sort of thinking about how we go about understanding genetic architecture, understanding selection on genetic architecture, and how that connects with phenotype, and eventually how does that invoke solutions about major problems. And so it's more the process interested in rather than sort of the end result. And none of us is wedded to any particular answer on these questions. So I'll stop there, and thank you again, Charlie, and in particular Meredith for organizing this wonderful event.
Okay, thank you guys so much. As I said at the beginning, we really try to have Club EvMed spark new conversations, new perspectives, and I think this is a perfect example of that, with so many different perspectives coming in from different fields and different backgrounds. And so thank you guys very much. Congratulations on a great paper. I think it's really, pushing the field forward. And I think we can see from the conversation today, there's so much more that can be done, and really even building a lot on what you guys have done. So congratulations on a great paper. Thanks so much for spending the time with us today. Thank you, Meredith. As Antonis said, thank you very much. And thank all of you for joining. It's great to see so many people here today and engaged with these conversations. Now, before we totally tune out, I want to show you the next Club EvMeds, at least the next few. We have, hopefully you can see my screen. We have Dan Lieberman next week, also Thursday at 12. These are not always at the same time and day, but it turns out we've got 3 in a row now on Thursdays at 12. He's going to be talking about a book that he has coming out. Title is Why Sitting is Not the New Smoking, an evolutionary medicine perspective on physical inactivity. I think he's going to make me feel guilty about my standing desk that I'm standing at right now. Then we have a Club EvMed Student Spotlight, where we're going to showcase 3 different graduate students in the research that they're doing. And we're going to have some breakout sessions at the end of that so that you can interact with those graduate students. And then 3 weeks out, another Thursday at noon one, I see, we have Joe Graves, who will be talking about everything you wanted to know about race and why that's important for evolutionary medicine, again, related to a book that he has coming out. And you can see it goes on. I'm not going to go through the whole list of things, but we have a great lineup, and we're all really excited to be able to showcase these events. So with that, thank you everyone again for showing up and engaging so much in this conversation, and we hope to see you next week and in the weeks to come. Thanks very much.
Thank you, everyone.
Thanks all.